Our cells have developed complex molecular systems to identify and repair lesions to the DNA molecule. When an alteration in the sequence or in the structure of DNA occurs, a series of coordinated events called DNA damage response happens. In most cases, sensors are able to identify and bind the DNA lesion and then to recruit other factors and create macromolecular complexes. These complexes can even be visualized under a microscope as small spots called foci. These macromolecular complexes trigger a series of coordinated events that result in the repair of the damaged DNA. The DNA damage response is regulated to make sure that the right factor comes at the right moment and place. During evolution, mammalian cells have evolved several molecular mechanisms to face different types of DNA lesions. If a single strand break occurs, a specific system called Bayer, base excision repair, is activated. In this case, some factors remove the portion of damaged DNA and then other proteins fill in the correct sequence. If certain chemical groups bind DNA, the Bayer system is activated as well. Thymidine dimers induced by UV rays are repaired by another system called NER, nucleotide excision repair. This system senses distortions of the DNA helix, removes the damaged portion, and substitutes it with a new one. When double-strand breaks occur, the cell repairs the damage using mechanisms of homologous or non-homologous recombination, depending on the specific phase of the cell cycle. Some errors in the nucleotide sequence could occur even during DNA replication. These errors are repaired by the mismatch repair system. These systems, as well as others, are important to preserve genome integrity. That means to keep the sequence stable over time. When any of these systems or factors are mutated, there is a greater risk of developing some types of cancer, since errors are not repaired. Some of these factors are also mutated in genetic diseases. To protect the DNA, cells have evolved different mechanisms referred to as DNA damage response.